In the 1830s, an English naturalist named Charles Darwin hopped on a boat and looked at some finches and figured out evolution. Around the same time, an Austrian monk named Gregor Mendel mated a bunch of pea plants together and figured out genetics and inheritance patterns. Evolution and genetics are now a staple of biology, confirmed by the discovery of DNA and the field of molecular genetics. We know that genetic traits that help an individual reproduce in their environment get passed down to their children, while traits that are harmful in their environment become less common as they have fewer opportunities to mate. Survival of the fittest by natural selection. These genes are seen as fixed, that you are born with all the genetic traits you will ever have, and traits that you gain throughout your life will not get passed down to the next generation. But is that really true? This idea was not always so obvious. Jean-Baptiste Lamarck was a French naturalist who came up with a theory of evolution before Darwin. Instead of believing that inherited traits were fixed, he believed that traits that changed over an individual's lifetime could be passed on to their children. The famous example is the giraffe. In Darwinian natural selection, some giraffes are born with longer necks than others, and those that survive and reproduce are those that are able to reach the treetops and eat some tasty leaves. In Lamarckian evolution, giraffes born with shorter necks can stretch and strain their necks trying to reach the trees, and develop the trait of a longer neck, which can be passed on to the next generation. Lamarck was not the first person to believe that traits developed over a lifetime were inherited. In ancient Greece, Hippocrates, the quote-unquote father of medicine, the Hippocratic Oath guy, also had some notion of inheritance of acquired characteristics. Hippocrates described an ancient tribe of people he referred to as the Macrocephali, meaning large-headed in Greek. This was a tribe of people who bound hard pieces of wood to the head of a developing child's skull while it was soft, so they would grow a long oval-shaped head. It is reported that Hippocrates said, "The characteristic was thus acquired at first by artificial means, but, as time passed, it became an inherited characteristic and the practice was no longer necessary." According to Hippocrates, after many generations of head binding, the macrocephali started giving birth to egg-headed children. The practice of binding heads and shaping skulls did indeed happen, and is a well-documented practice in many cultures across the globe. Archaeologists have actually found the skulls of people with oblong heads, and the head binding practices have been documented well into the 1900s. Because of their long, oval shape, these findings are often the subject of speculation in ancient alien circles. Similar to Chinese foot binding and European corsets, head binding, or artificial cranial deformation, is sometimes used to demonstrate status. Some members of the Chinook tribe of the Pacific Northwest had a similar practice of head flattening for social status by tying a board to a baby's forehead while they grew. In Stalinist Soviet Russia, a biologist named Trofim Lysenko proposed a method of crop rearing that largely rejected Darwinism and Mendelian genetics in favor of a form of Lamarckism. Lysenkoism was promoted as a revolutionary agricultural strategy to increase crop yields after a devastating famine. The seemingly competitive nature of natural selection was seen as capitalistic and at odds with the prevailing ideology. Genetics was declared a bourgeois pseudoscience, and many biologists were persecuted under Stalin's rule. Lysenkoists claimed that they could vastly improve agriculture and turn rye into barley and weeds into crops by altering temperature and humidity alone. Unfortunately, these practices did not transform one species of plant into another. Lysenko's methods failed to dramatically increase crop yields, and he eventually fell out of favor. Nowadays, Lamarckism is often presented as a failed theory of evolution. Lamarck was certainly a smart cookie, but then Darwin and Mendel came around and made Lamarck and everyone else on Earth look like a bitch. But what if he was onto something? You might have heard that every cell in your body has the same DNA, the same genetic sequence. And that is true, except for minor inconsequential mutations, and B-cell antibody genes, and red blood cells which don't have any DNA, and sperms and eggs which only have half the DNA of body cells, and the genomes of the bacteria in your gut, which I guess you could consider a part of your body, and the off chance that you have genetic mosaicism or tetragometic chimerism or an organ transplant, and of course all the mutations in cancer cells. But ignoring those exceptions, it is by and large pretty much mostly definitely true that all of the cells in your body have the same DNA sequence. A brain cell has the same genetic code as a muscle cell, but they do different things. You can't make a brain out of muscle cells, or a muscle out of brain cells. Genes are regions of DNA, regions of your chromosomes, that code for proteins, and proteins give a cell its function and shape. So why would cells with the same genes have different functions and shapes? All cells have the same genes, but in different cell types, different genes are turned on or off. If a gene is transcribed into protein in a cell, we say that gene is expressed. Different cell types express different genes. They make different proteins. One way that gene expression is regulated is by proteins called transcription factors, molecular factors that turn genes on or off. Transcription, as in reading a gene to express it, transcribing a gene to be expressed as protein. Some transcription factors cause a gene to be expressed more, while others repress a gene's expression. These transcription factors ride the DNA strand of a chromosome and attach to a specific DNA sequence adjacent to the gene they repress or excite. If they excite the gene, the transcription factor helps stabilize and attach the molecules involved in the transcription of the gene. They let the proteins that express genes attach and read the gene. If the transcription factor represses the gene, it blocks proteins involved in reading the gene. The actual sequence of the DNA, the code of the gene, is not altered in any way. Transcription factors are themselves proteins, and like all proteins are coded for by genes. During development, signals from neighboring cells bind to these factors and alter their shape, which can cause them to bind to their target genes and alter expression, or can cause them to be unable to attach to DNA at all. This makes it so that different cell types express different genes, because they have different transcription factor activity. Different genes are excited or repressed. Transcription factors are not the only proteins that interact with DNA. DNA is wrapped around proteins called histones, molecular spools that the chromosome coils around. These histones can be modified to wrap DNA more tightly or loosely. Genes on DNA that is wrapped tightly around histones are less likely to be expressed, since the proteins that read the genes are less able to access them. Loosely wrapped DNA exposes the genes to be read, allowing the gene to be expressed more. Different cells have different genes exposed. Different regions of DNA will be wrapped tightly, and this is another way that cells control their gene expression. The DNA molecule itself can also be modified to turn genes on or off, without altering its sequence. Proteins can attach chemical tags to DNA called methyl groups that typically silence the gene they methylate. These methyl group tags do not alter genetic sequence. The sequence of A's, C's, G's, and T's in DNA does not usually change when modified by repressing transcription factors or methyl groups. The code is still there, it is just no longer accessible to the molecular machinery that reads genes and makes proteins. Among other processes, transcription factors, histones, and methylation are the reason why cells with the same genes have different functions. It is because different genes are expressed in different cell types. Many of the genes for muscle function are inaccessible in a neuron, while the genes for brain function are repressed in a muscle cell. Environment can also affect gene expression. Substances you are exposed to, stress, nutrition, lifestyle choices, and experiences throughout your life can change the expression of your genes, turn some genes on and others off by activating or inhibiting transcription factors, altering how tightly wound regions of DNA are, or by causing the methylation of certain genes. If you have an identical twin, you may have the same genome, the same DNA sequence, but differences in your upbringing and life histories will make you express those genes differently. In some of your genes, some will be turned off and others turned on in your twin. In simpler terms, genes can be turned on or off, often in a way more like a dimmer switch than a binary on-off switch. Different cell types have different genes on or off, and cells adapt to changes in the environment by turning certain genes on or off. Methylation of a gene often turns that gene off. It has been demonstrated that exercise can change your genetic methylation patterns. In a study of 23 men with no previous history of regular exercise, Swedish scientists found that 6 months of an exercise program changed the methylation patterns in the fat cells of the men. After exercise, the men had more methylation of many genes in the fat cells, and found alterations in the methylation of genes involved in type 2 diabetes and obesity. This suggests that exercise can help prevent illness by turning genes on or off. The growing science of epigenetics is studying the molecular mechanisms involved in gene expression, how these modifications change, and to ask to what extent these modifications can be inherited. Epi, meaning on top of, and genetics, of course, referring to genes. Epigenetics studies the histones, methyl groups, and other factors that regulate genes. What if these factors can be inherited? Could the actions you take that alter your epigenetics be passed on to your children? In order for an epigenetic mark to be passed down, it would need to occur in a sperm or egg cell, the cells from your parents that become you. In many animals, there is a phenomenon called genetic imprinting, which has nothing to do with the baby ducks following their mother after hatching. That's a different kind of imprinting. Erase that from your brain. Genetic imprinting is an epigenetic phenomenon that causes the genes to be expressed in a parent-specific manner. Epigenetic markers, like methylation tags, from the parent get carried into the womb, causing expression or repression of certain genes involved in fetal development. Markers from the father's sperm tend to be growth promoting, whereas markers from the mother's egg tend to be growth limiting. Sperm provide the DNA with methylations repressing slow growth, while eggs provide the DNA with methylations repressing fast growth. Evolutionary biologists argue that this evolved because males want their progeny to be robust, to grow quickly, so their genes can pass on optimally. If you are a male, your children are more likely to pass on their genes if they come into this world big and strong, and it doesn't necessarily matter if that destroys the mother's uterus in the process. The genes of males with well-developed, robust children are more likely to survive. Meanwhile, the factors from females limit growth so they can reproduce again in the future, so their reproductive organs aren't damaged during development of the fetus or during birth. The genes of females who can mate and give birth multiple times, females that don't have busted uteruses, pass on their genes more often. This creates a sort of genetic arms race between the DNA of the mother and the father in the developing fetus. In pair bonding species, species that mate for life, this genetic imprinting phenomenon is not as common or as intense. So, there are parent-specific traits that are inherited, but what about your parents' experiences? Could that affect development? During World War II, there was a 3-month period in the Netherlands when the Nazis diverted food away from the Dutch, causing mass starvation and death. This period was known as the Dutch Hungerwinter. People who were a third trimester fetus during that time got signals from the mother that food was scarce, and when they grew up had 19-fold higher rates of insulin release, obesity, diabetes, and hypertension. The starvation did not mutate their DNA, yet their children inherited the consequences of the famine. This suggests epigenetic factors led to metabolic programming to cope with the famine. Somehow, the Dutch mothers sent biological signals to their children that there was little food in the world they were being born into. This turned on genetic metabolic processes that would help them survive a famine, but led to metabolic diseases when plenty of food is available. Their bodies were set up to prepare them for a world in which they would experience starvation, but would end up being detrimental in a world with food available. But we already know that pregnant mothers can influence the development of their children. That's why we tell pregnant women not to smoke or drink or get punched in the womb. But could experiences before conception affect the development of the child? War, famine, death, plague, and other horrific events can cause lasting trauma in the people experiencing it. But that trauma may get passed down to children and grandchildren as well, even if they did not experience the events themselves. People who are the children of Holocaust survivors have a higher rate of PTSD than the average person, even if the children themselves never experienced the trauma. In 2015, a study looking at 32 Holocaust survivors and the 22 children of Holocaust survivors found what may be an epigenetic basis for inherited trauma. The survivors and their children both have methylation on a gene involved in stress hormone levels. The gene codes for a protein that makes the receptor less sensitive to stress hormone, less able to bind and be activated by the hormone, meaning the stress hormone becomes less effective. The methylation of that gene would inhibit the formation of the stress-repressing protein, which may explain the higher prevalence of post-traumatic stress disorder. This pattern of methylation in survivors and their children may contribute to their psychiatric stress-related illness. It is very important to note that this study has a very small sample size, and only 9 control subjects, 9 Jewish participants who were not children of survivors. Those who were children of survivors were raised by their biological parents. Intergenerational trauma is a well-established phenomenon, but this study alone does not necessarily prove that the trauma was inherited epigenetically. It could have been passed on environmentally. Survivors may have shared stories of the genocide with their children, and the traumatic events that they lived through may have had an impact on their parenting style. The methylation patterns they share with their children may be the result of a similar upbringing, a pattern inherited by grandparents before the Holocaust, or perhaps trauma induces these epigenetic markers whether the ordeal is experienced firsthand or through the stories of loved ones. Knowing that your parents, people you love, went through a horrible tragedy might be traumatic to hear as a child, even if you didn't experience it firsthand. In the American Civil War, many Northern Union soldiers were kept in harsh, overcrowded POW camps by the Confederate South. It was later found that the sons and even grandsons of these underfed prisoners of war had higher rates of mortality than those whose ancestors were non-POW veterans. Like the children of Holocaust survivors, these POW sons and grandsons never experienced the camps firsthand. What is interesting about this case is that the higher mortality trait seems to be Y chromosome linked. The daughters were unaffected, and the sons conceived before the POW experience are also unaffected. This suggests that the trait is not a result of genetic sequence, because if the trait was conventionally genetic, pre-camp sons would have the same risk of death. The researchers who published these findings suspect the mortality is due to inherited epigenetic markers that formed during the POW's trauma, although they haven't identified the specific methylations or histone modifications involved. It is also possible that the Union POW veterans raised their sons different than their daughters, and that sons born before the camp experience were too old to be affected negatively by the changes to parental behavior. While these examples are a bit grim, there are also cases of beneficial inheritance. In the Swedish village of Överkalix, the local church has kept a thorough record of town members and annual crop yields. Turns out, sons and grandsons of Swedes who lived through low crop yields and grew up underfed had about a 1/4 lower incidence of developing heart disease and diabetes. That is, specifically if the grandfather lived through a crop shortage when he was between 9 and 12 years old. Sons of hungry Swedish tweens were found to live on average 30 years older than the people who had fully fed fathers. Once again, if this is an epigenetic phenomenon, we don't know the mechanism. We don't know which genes got repressed or how it got passed down. It also seems at odds with the Dutch hunger winter babies. Why do the children of malnourished Dutch mothers fare so poorly, while famished Swedish fathers have healthy, happy sons? And what about memories? Maybe you can inherit different sensitivities to diseases like PTSD or heart disease, but can you inherit memories? A learned experience? The science of memory is not fully understood, but we know that memory formation and processing depends on the synapses. Synapses are the connection points between neurons, brain and nerve cells. These are gaps between cells where signaling molecules are released from one neuron to another, either to activate or inhibit an electrical signal in the postsynaptic neuron, the next cell. Brain cells create a network of electrical connections which form the basis of every perception, emotion, thought, behavior, and memory you experience. It is in the structure of that network, the strength and configuration of the connections between brain cells, that memories are formed. If one neuron often signals to another neuron, this strengthens the synaptic connections between the two neurons, by inducing the formation of more signal receptors, causing the release of more signal, and by growing new synapses, more points of connection. Cells that fire together, wire together. This is why practicing, spaced repetition, and other activities that involve hearing and doing things a bunch of times will help you remember them, because it keeps activating the same neural circuit, activating the same brain cells, which strengthens the connections between them, making them more likely to fire together in the future. New memories are likely formed by the formation of new synapses, new connections between neurons. But what if there is more to memory formation? What if there was a chemical basis of memory, an engram that could be transferred from one person to another? James McConnell was an American biologist who proposed a theory of memory that suggests long-term memories are stored in molecules, such as RNA. RNA are molecules very chemically similar to DNA, although they are less stable and they are more versatile than DNA. In order for a gene to be translated into protein, an intermediate RNA molecule is formed with a copy of the genetic information, a complementary sequence. This acts as a messenger to the molecular machinery that reads the RNA and translates it into protein. But not all RNA is coding. Many RNA molecules in the cell never turn into proteins. Many RNA molecules have functions. They facilitate important processes in the cell. McConnell hypothesized that there are some molecules that can facilitate memory, perhaps some form of RNA molecule, and that those RNAs can be transferred from one organism to another. This theory proposes that in addition to synapse formation, or instead of it, memories are formed epigenetically by RNA expressing or exciting certain genes or processes in the cell. This is based off of an experiment where flatworms, called planarians, learned to solve a maze, were ground up, and fed to untrained worms, which were then able to solve the maze more quickly. Planarians are small worms with simple brains, and are able to learn simple tasks like solving a maze. Proponents of the memory RNA hypothesis suggest that when worms learned the maze, the memory was stored molecularly in the sequence of an RNA molecule. When the worm mulch was cannibalized, the next planarian inherited its meal's memory of the maze. This experiment hasn't been replicated. Another researcher has not been able to see the same result doing the same experiment, so the hypothesis is far from confirmed. As an aside, Dr. McConnell was targeted by the Unabomber, a Luddite domestic terrorist who sent a mail bomb to his house. McConnell survived but was deafened by the attack. Another more recent study suggested that injecting the RNA of a sea slug that was periodically shocked into an unshocked slug could allow it to learn the response behavior of the donor shocked slug. When given a mild shock, a sea slug will briefly withdraw its siphons and gills into its body to protect them. When the slugs are shocked often, they learn to withdraw their siphons and gills for a longer period of time. They learn that once they are shocked, they may be shocked again, so it pays to keep their giblets protected. The unshocked slugs that were injected with the shocked slug's RNA withdrew their gills for long periods of time in response to the shock, while unshocked slugs given unshocked slug RNA withdrew briefly. This could suggest that something in the code of the RNA allows it to interact with genes in the snail's neurons, telling it to expect another zap and to protect its gills. The RNA could simply be a response to stress, which turns on genes or factors involved in long withdrawal, and is not necessarily an implicit chemical memory engram. It may even be a stretch to refer to these as memories. Instead, they could be the chemical basis of a reflex. There is no evidence that the sea slugs or worms consciously experienced the explicit memory of the donor organism. Imagine we did an experiment where we scared somebody and induced a panic response. They would have elevated stress hormone levels. Then, let's take a blood sample of that person, isolate the stress hormone, and inject it into another subject. If that subject also has a panic attack, that doesn't mean they remember the event that scared the previous person. It just means that the first subject's hormones put somebody else in a physiological state of panic. In the example of the cannibalistic planarians, perhaps the worm smoothie didn't even give any information about the layout of the maze, but maybe the worm synthesized a signal that boosted brain function or problem-solving skills over the course of learning the maze, that the second worm then absorbed. Unfortunately, we don't yet have the technology to talk to worms, so we can't be sure they are truly experiencing a memory of anything. The idea that memories are stored chemically or epigenetically in RNA is not as robust or well demonstrated as the model of memories being based in synaptic connections between neurons. But these are worms and snails. What about complex galaxy-brained organisms like us? Show me an inherited memory in something that lactates. Well, male mice were taught to associate a fragrant, fruity scent with a mild electric shock, and developed a fear response to the sweet scent. After internalizing that the scent was coupled to a shock, the mice tensed up in a Pavlovian response to the smell, even when they weren't shocked. Unshocked mice have no problem with the smell. They don't tense up when the odor is present. Surprisingly, the children of shocked mice also exhibit a fear response when exposed to the scent, even when they themselves were never shocked. This fear response was likely encoded by epigenetic markers on the mouse's DNA in neurons, so it would associate the scent with danger every time it encountered it. Then, somehow, this epigenetic information got passed down to their children through the sperm. This fear response is passed down to the mouse's offspring even through in vitro fertilization, meaning that the response was inherited even though the father didn't directly interact with the mother or the child. The young naive mice never met their fathers, so their mouse dad couldn't have warned the children to watch out for an evil fruity smell. No one told the child mouse to expect a shock coupled to the scent. Looking at the genes of the shocked father's sperm and the unshocked children's sperm revealed a loss of methylation at the gene for the receptor for the fruity scent. Loss of methylation means less repression of the receptor, meaning that the mice make more of the receptor, meaning the mice would be more sensitive to the scent, which would make sense that you would want to be more sensitive to a scent that may signal harm will come your way. So, we see that the fear of the smell was learned at a molecular level in the fathers, and that learned molecular mark was passed down to his naive children. If the methylation loss only happened in the mouse's olfactory cells, only in the cells involved in smell, they would not have been transferred to the children. Somehow, his sperm also learned the fear response to the smell, and that allowed the fear to be inherited. We don't know how this trait got into the sperm, how the smell receptor gene got demethylated in the sperm DNA, or how this methylation pattern transferred to the children. So what do we make of all this? Does this mean that Lamarckian inheritance of acquired characteristics plays just as big a role in evolution as mutations to the genetic code? Without understanding how it works, the idea of transgenerational epigenetic inheritance can be used to justify many ideas. Some will claim that you can inherit literal memories, which are either manifested in subtle behaviors and perceptions, or full-fledged memories sitting dormant in your mind, waiting to be unlocked by some practitioner of past life regression therapy. Others may claim that the strikingly similar stories across cultures are actually the inherited memories of our ancestors. Many cultures have myths about a great flood precisely because we all have some latent memory of a gigantic flood that really happened in our shared ancestors' past. Epigenetic inheritance is still a brand new field, and the only thing we can say for sure at this time is that we don't know how it works or to what extent these traits are passed on. Any claims of shared ancestral memories or latent past life experiences cannot be justified by epigenetics alone, just yet. And there's a huge confounding factor with the idea of transgenerational epigenetic inheritance in mammals. As sperm and eggs are formed, almost all of their methylation tags are erased, not only then, but also during the early stages of the embryo's development. In our earliest embryonic stages, we are balls of stem cells that can become any cell type. Epigenetic marks, like methylations and histone packaging, turn genes on or off to determine what function a cell will have, if it is a blood cell, or a neuron, or a skin cell, or, a muscle cell, etc. In our sex cells, these marks are removed, so the resulting embryo starts out with a clean slate. Inheritance of DNA methylation patterns is presented as one of the most likely ways that these epigenetic traits are inherited, from transgenerational trauma to the fear of fruity fragrance. But this intergenerational reprogramming seems to challenge our mechanism of epigenetic inheritance. If the marks gathered during a lifetime are largely erased between parent and child, how does the child inherit them? What about the phenomenon of genetic imprinting, where methyl groups inherited from the father promote uterus-busting growth, while the methyl groups from the mother limit growth in an attempt to foster quantity over quality? Somehow these imprints, methylation patterns involved in embryonic growth, are often spared from demethylation in most cells. The epigenetic tags from each parent that limit or promote growth don't get removed. Exactly how this happens, how their cells make sure these marks don't get erased, is a bit of an unknown. Overall, it seems that some epigenetic marks are spared, and it is possible that the inheritance we've seen in these studies are due to the lucky few marks that do not get erased during early development. For humans, it is difficult to say with any certainty that a trait is epigenetically inherited, that any characteristic is due to events that transpired during the lifetime of a parent. A trait could be the result of parenting or similar environment, or due to a mutation to the genetic sequence passed down to the child. So, to reiterate, before genetics was well understood, many believed that experiences and traits acquired during life could be inherited, from Hippocrates' description of inheritance of elongated skulls after head binding, to Lamarck's suggestion that giraffes evolved by inheriting the longer necks of those who stretched to reach tall branches. For the most part, every cell in your body has the same genes, but express those genes differently to make different cell types. Molecules that interact with DNA can turn some genes on or off, and are influenced by the environment. These changes to gene expression are called epigenetic marks. In some tournament species, epigenetic marks from sperm cause the embryo to develop quickly, at the expense of the mother, while epigenetic marks from the egg cause the embryo to develop slowly. There is evidence that trauma can be inherited, causing health effects in your children and grandchildren. It is difficult to say if this is because of culture and parenting, or if epigenetic marks that alter gene expression are inherited. Are the inherited health effects due to nature or nurture? In laboratory experiments of worms, slugs, and mice, it seems that memory can be transferred from one organism to another, or even inherited transgenerationally. But it may be too sensational to refer to this as a memory. We have no direct evidence that the mice or worms experienced their donor's memory of the event. Instead, all we can say for sure is that we can transfer physiological responses like stress and fear between animals. To what extent epigenetic changes can be inherited is still an unknown, but it is still worth living your life as if your actions will be inherited by your children. So get good nutrition, exercise, socialize, sleep, and yada yada whatever. If it doesn't help your kids, it'll still help you during your own life. On the flip side, don't worry so much about dooming your children if you ever had a bad experience. The field of epigenetics also shows us that human beings are resilient and adaptable, and that sometimes even quote-unquote bad genetics can be overcome by positive environmental input. Okay, well, that's about it. Have a good night, folks. Bye.